All right, folks, so I want to show you what your notebook needs to look like. We're gonna put a whole bunch of pages together into the notebook, and I wanna make certain that you are super set up for where we're gonna go when you guys get back from spring break. So I know that spring break is exactly that, a break. However, you've been doing really, really well with your current work and you're starting to come up with a nice pattern, and I'm pretty sure you're not gonna go too far for spring break. So I want to show you this. I'm going to go take a quick spin through our notebook, and then I'll explain a couple of pages with our next video. So here we are on page 69. This is just your cover page. It was a flat mount. And then we went over to 70. This was your targets. I think if you read through your targets, we are pretty close in here, including the replication. And now we're going to get into cell cycle here when we get back from spring break. The next page is going to be your What is DNA? That's page 71. And then we put together just a little bit of notes from the first two tasks that we did on day one, the what we know from learning genetics and what are genes, and then the textbook work. So this was my version. You don't have to have the exact same thing as my version, but this was my version of my understanding about DNA based on the 2 or 3 things that we had done at the end of day one. So moving on, that was page 72. 73 was your drawing that you did your Flipgrid explanation on, which was really good. And then a little bit of practice. There was a detailed video about all the details down in here. And if you don't have that in your notebook, then I would like you to go back. I don't know which day it was, but go back and you'll see the video of me explaining the DNA detail worksheet here and explaining your patterns that you're seeing for each side and the idea of complementary and anti-parallel. Next, we'll go on to page 75. Page 75 was also a flat mount. This was our DNA structure POGL. The first 2 pages are DNA structure on model 1. We had our nucleotide, we had our ladder model, and our real twisted helix model of DNA. And we did both of these pages with nice flat mounts, 75 and 76. And then you went to the model building. You put together a big, beautiful model and then shot me a photograph of it. This is your page 77, which looks great. So now we're going to dig into some of the newer pages that I'd like you to add. The next page you're going to add is going to be a hinge mount. So we're going to mount it on the side. I always cut off a little bit on the inside so the page is more narrow. But this is a hinge mount of a DNA practice sheet. This is on 78A, and then 78B is on the inside with your DNA song. Of course, you would love to sing that a little bit over spring break. Who wouldn't want to sing on spring break? And then there's one more page, and I cut this one way down because it was a little bit too big, and it had a picture or a coloring assignment on the back that we're not going to do. But this is a great practice page, again, for DNA's structure on 78C. So that was a hinge mount. We'll go ahead to page 79. 79 was what we just did on Thursday with DNA replication. So we're talking about how we can get really perfectly copied DNA in every single one of our cells. When we started and we were conceived, we were just a mere single cell with 46 chromosomes in that particular cell. And so we have turned into these complex organisms that have trillions and trillions of cells, and each of them have their own full set of DNA, the exact same DNA that you had on the day that you were conceived. So we went through the process. We went through helicase, breaking open the double strand, polymerase, adding new nucleotides. The new nucleotides that were being added are blue, and then the final bit of proofreading that happens with DNA polymerase, and then ligase filling some gaps, and we end up with 2 exact copies of the original DNA. The original DNA doesn't exist anymore. Half of it's right there and half of it's over here. But what we have is the exact same pattern and order of nucleotides. So that was page 79. Then we're going to add 80, 80A and B. This is a hinge mount. I'd like you to answer these questions, but I'll tell you later on which pages need to be completed. This page is a great practice, and it's a continuation from the POGIL that started with DNA structure. So we'll do front and back there. And 80C is blank. Then we're going to leave a couple pages blank in the middle. So we're going to leave 81 blank and 82 blank. And then we're going to get going to our cell cycle work. Page 83A is a hinge mount, and it's got the cell cycle and stages of mitosis on the back side, and mitosis in the cell cycle in the front. And then we're going to do a mount. You have already seen this sheet before. On this side there was a dotted line, and we had cells here, and I asked you to cut them out and not lose them. So still keep that little packet of cells someplace close by, and then I want you to cut this and tape it down, but tape it so that you can fold the bottom up, because this is going to be long. Okay, so that goes on page 84. Now there are two cell cycle processes. One is mitosis, and then the next one is meiosis. So the meiosis is going to come very next set of pages. This is 85A. We're going to do another hinge mount with this page, and on the back it's going to say 156. That'll be your 85B. And then you have another sheet. It's a very small sheet. It's got really little teeny tiny cells, and you'll need to cut it out. Again, I'll show you this shortly, but what I do need you to do is go grab yourself a piece of printer paper. I want you to fold it in half, and then I want you to cut it so that you can tape the printer paper together and tape it right at the bottom there, because we're going to do another cell cycle, but this one is even longer. So we've got to make room for that. Okay, so please make certain that you prep this up. This is on page 86 of your notebook. This is going to be for our meiosis process. All right, almost done with this. Page 87, I'd like you to set up. This is going to be a comparison, a big Venn diagram. We're going to put cell cycle with mitosis information on this side, cell cycle with meiosis on this side, and then this is what's going to happen in both, what these two processes have in common. You don't have to fill anything in yet. I just want you to set the page up. So please make certain that you set it up. I would love some color, but if you can't do color, all pencil will be just fine. And then on 88, this will be another blank page for a little while, but we are going to add some additional cell cycle details on page 88. All right, your last pair of pages. The last two pages I'm going to ask you to prep up are going to be 89 and 90. So 89 is an explanatory page. It's a hinge mount, so we're going to do a very careful hinge mount job on this. When you tape it, make certain that you're careful you don't tape other pages together. But when you tape this, we've got to be able to see the front that has something called a karyotype, and then a pregnant mama's belly, and on the back there is a big grid, a table of genetic anomalies, and then this page is going to be blank. That's 89A, and then this is a little bit complicated as well. There's going to be a few pieces on this page. So the first page is identifying karyotypes. I cut a little bit off of each side of this page, so it was really a bit narrow, because it's going to be the top of my page 90. Underneath it, so this one's got two pages, underneath you have a sheet that says a karyotype form and then a spread letter spot. It's got a whole bunch of places here. This is a flat mount. This goes flat on page 90. And so this will be 90C. Then the back side is 90B. And when we have this hinge properly formed, that will be your 90A. So that's your notebook at this time. I'm going to go ahead and post which pages need to be completed. Then you're going to take a quiz on DNA replication, and then I'm going to ask you to complete just a little bit more prep work for when we come back from spring break. Take care.